everyone to our second webinar. We are very happy to share this meeting with you. Again, each seminar consists in 2 research talks, and each speaker will deliver a 30-minute presentation with an additional of 5 minutes. And there will be 20 minutes in the end for a final discussion. We hope you have the opportunity to interact with our guest speakers and ask them lots of questions. Today, we will be learning about integrated pest management, and that is what's part of the presentation. And the first talk is titled Seeking of Host Plants by Chemical Cues in Herbivore Insects by Dr. Patricia Fernández. She is a CONICET researcher and a professor from the Agricultural School of University of Buenos Aires. She holds a PhD in chemical ecology, and she works with plant-insect interactions. She studied volatiles and other secondary metabolites associated to leaf surfaces that can affect oviposition and or feeding choices, and how these cues change after insect damage. The second talk is titled The Cereal Aphid Manager App by Dr. Tyler Wist. He works as a field crop entomologist with Agriculture and Agri-Food Canada to understand the effect of crop ravaging insects on their host plants. He spent a lot of time in wet and pulse crops these days, looking at plant resistant to weed mitch and insecticide timing for aphids, and still take walks through cereal crops to check on cereal aphids and their natural enemies. So we are going to start with Patricia Fernández.
Hey, hello. Okay, hello. Thanks, girls, for the invitation. I was really happy and to finish this crazy year with this talk. I'll share screen, okay?
Yes.
Sure. I hope it will screen. Okay, tell me please if you can see it at some point. No, I'm not selected. Okay.
We can see your PowerPoint.
Okay, perfect. Okay, so this is my talk today. The name is Seeking of Host Plant by Chemical Cues in Herbivore Insects. My name is Patricia Fernández, and I work at the University of Buenos Aires and CONICET. Well, as you know, the interaction, I need to let me do this. Okay. The interaction of plants and the herbivorous insects has shaped the evolution of an intricate network of defenses and counter-defenses for millions of years. The result is a surprising diversity of phytochemicals and plant strategies to grow and survive. And yes, trees are specifically challenged to resist the plethora of biotic and abiotic stresses due to their dimension and longevity. And specifically about phytochemicals, they are typically exploited by highly specialized herbivores as cues for host orientation. Imagine for a tiny insect, a treetop is not an homogeneous, predictable habitat, but rather an heterogeneous and often inhospitable environment. So, during host seeking, an insect starts using volatiles and visual cues in a range, and then once she arrives to the leaf, she will use the chemical profile that determines the final acceptance. So the phytochemistry can affect site selection for feeding and also oviposition. Well, this is a study area. Here we are in South America. This is Argentina, and here we are in Buenos Aires. This is the delta of the Paraná River. This is the biggest wetland in Argentina. It possesses a high biodiversity and unique ecological conditions. And here, willow plants have a potential of development and became highly relevant for the economy of the area. And this is the study system. The willow from the genus Salix The family is Salicacea, and they are bred here for food production or genetic improvement. That is why we have available a lot of different genotypes. And we also have the willow sawfly, Nematus oviposition, and this is a main willow pest. So I'll talk a little bit more about the willow sawfly. It is a highly specialized sawfly feeding on selected willows. Females are parthenogenetic, so they don't seek mating partners. Adults don't seek for food or conspecifics. Actually, as soon as they hatch, females look for oviposition sites. So this specialization for egg laying suggests a sensory system which is highly tuned for recognition of suitable oviposition sites. Okay, so the goal of this study, the main goal, is to study orientation cues used by females to seek and accept the host plant in order to connect preference with plant phytochemistry. And this will allow us to recognize susceptibility factors in willows that favor host finding by pest, which can be important during genetic breeding. So these are all specific objectives, and I will take you through all of them, yes, along the talk. The first one is to determine feeding and oviposition preference in the field and in the lab. So this is how we started this job. With a field survey, Teresa Cerrillo, she studied the feeding damage of different genotypes in the field. These are different genotypes, and here you can see the level of damage. And she found that some genotypes, like Salix viminalis, had very, very little damage, and some other ones had a lot of damage in the field. So then Selina Braccini, as part of her PhD thesis, she did a multiple choice bioassays by analyzing oviposition, and what she found, so these are all the genotypes, and this is the number of eggs laid. She found that also this genotype, Viminalis, received few eggs, and the Nigra genotypes received most of the eggs. So we said, okay, Salix nigra genotypes are most preferred, Salix viminalis genotypes are less preferred, and the feeding preference is, at least in part, determined by the oviposition preference. So to continue, we studied the role of volatile cues in long-range orientation. So we did bioassays by Y maze. This is a Y olfactometer. We offer on one side a Salix nigra branch, and on the other side the Salix viminalis. I will use red for the non-preferred, so you remember, and I will use green for the preferred one, and then you release the insect in the main arm. What we found was that for the preferred genotype, so the preferred branch, the females choose them first, and also they spend more time in that arm, and the contrary with the less preferred genotype. So we think from here that willow sawfly is orientated to volatiles of the preferred genotype Salix nigra. So then we continue and we analyze the chemically the volatiles. This is how we do the volatile analysis by headspace and then GCMS. This is the profile of a nigra plant, and this is a profile of a Salix viminalis plant. And you can see the profiles a little bit different, and also there are some specific compounds for each species. These are all the compounds we found, and specifically for Salix nigra, they were, for example, osimen, undecan, and decanal, and then for Salix viminalis, the non-preferred, we have also only caryophyllene. So what we found till here is that females seem to orient to volatiles. These volatiles could be related to female orientation, and we know that once they arrive to the leaf, upon contact, leaf surface morphology and chemistry may offer numerous additional stimuli for host acceptance and recognition. What we did was to characterize the behavior during leaf surface recognition before oviposition. And now I'll try to show you a video in which you will see Yes, here. This is the main behaviors we analyzed. The antennation while walking. You can see that she's like looking with the antenna, and then she starts rubbing the leaf surface with abdomen. Yes, so we quantify these behaviors, and what happened? This is that the antennating while walking was similar for both genotypes. We did this with a petri dish, yeah, was similar. But then the abdominal sweating was really performed in more frequently in Salix nigra, yes. And then normally in Salix nigra also, after abdominal sweating, they lay an egg, and it didn't happen in Salix viminalis. So what we think is that the preferred genotype maybe possess contact cues that stimulate abdominal sweating and oviposition. So then we characterize the sawfly ovipositor and the egg deposition. For example, here is by a light microscope how an egg looks like. This is the egg in an electron microscope, and you can see that she doesn't damage the surface before laying the egg. Then this is a general view of the ovipositor. This is actually the saw, that's why the name sawfly. And then we also found that here in the circus, they possess chemosensory sensillas. And this is the cross section of a leaf blade bearing an egg. So this is the egg. And what she does is she normally with the saw make a hole and insert the egg like in a pocket. So with this, I want to show you that there's an intimate contact between the sawfly ovipositor and the leaf. So we estimate the importance of leaf epicuticular waxes for host recognition. In order to do that, we did bioassays by gamma arabic. Gamma arabic is a polysaccharide where you can paint, like here, you can paint the leaf and then peel and remove the waxes. So here you have two leaves. This one is without waxes and this with waxes. The interesting thing is that the leaf is alive, yes? And we offer them to a sawfly. These are the results. This is the number of eggs, and here you have Salix nigra and viminalis, yes? And if you see Salix viminalis didn't change, the number of eggs is always low, with or without waxes, but for Salix nigra, the sawflies laid more eggs in the in the leaf with waxes, yes? So the sawflies laid more eggs on leaves with their waxes intact. Salix nigra waxes are important, but not essential because we still found eggs here. And Salix viminalis do not possess repellents, at least in the epicuticular waxes, because when we remove them, they still don't lay eggs. Okay, so till now we can say that contact cues matter. Thus, we perform a chemical analysis of the leaf surface, and this is what we did. This is very simple. It's just solvent dipping, and we did non-polar extract by dipping 20 seconds in dichloromethane, and polar extracts by dipping 20 seconds in methanol. Okay, so this is the analysis of the non-polar extract. If you, if you see, okay, these are all the compounds we found. For Salix viminalis, you can see that you have mostly alkanes, yes? You can see them here, and these are the 4 alkanes we found, pentacosan, heptacosan, nona, and triacosan. So mostly alkanes, but when you go to the preferred genotype to Salix nigra, you can see that there's like maybe alcohols, acids, esters, so like more diversity of compounds. So cuticular waxes are composed mainly by alkanes, but preferred surfaces were chemically more diverse than non-preferred ones. We didn't find one differential compound, just this diversity. Okay, and then we did the analysis of the methanol extracts, and this was very interesting. We did it in Vienna with Michael Reichelt and Jonathan Gershenson. This was very interesting because we found that in Salix viminalis,
we had no phenolic glycosides that were present in Salix nigra. Salicin, salicortin, and tremulacin, these are phenolic glycosides, and these are typical compounds from willows, and they are only present in Salix nigra. So, preferred surfaces possess phenolic glycosides, which are absent in non-preferred ones. So, till now we have this high diversity of non-polar compounds in the boxes, and also the presence of phenolic glycosides. Our question was, it might be a fingerprint for them. Well, we know that insects use plant secondary metabolites as fingerprints to recognize their host and oviposit on them. And our hypothesis was that phenolic glycosides are part of a fingerprint that leads to oviposition in willow plants. And why phenolic glycosides? I think this is important. These are the compounds I'm talking to you: salicin, salicortin, and tremulacin. And these are the most abundant secondary metabolites in Salicacea. Actually, tremulacin and 2,0-acetylsalicortin have been proposed as oviposition cues for willow sawflies from another species, and also because we found a marked difference in these compounds between preferred and non-preferred willow species. So what we did was to connect oviposition preference with PJ levels. For that, we analyzed natural colonization, oviposition preference, and PJ content in different commercial genotypes, and then we correlated them. Okay. For the field damage, again, Teresa Cerrillo helped us. These are all commercial genotypes. And she did a field survey, and she studied the feeding damage there. Here's the level of damage, and these are the different genotypes. And again, Selina did the oviposition preference in the lab with these commercial genotypes, and she found that there's a clear oviposition preference, especially you can see it's easy to see in the lab. And then, so we can say that field damage is, again, at least in part, determined by the oviposition preference. So now, Camila Davila, which is another PhD student, she analyzed the oviposition preference in relation to PJ's concentration. She studied salicin, salicortin, tremulacin, and total PJs, and she analyzed the concentration as a function of oviposition preference. And what she found is that there's a positive correlation in all cases is significant. So we think that this phenolic glycoside seems to have a role in host selection. So we think that there might be a fingerprint for the sawfly related with these compounds. And this is interesting because secondary compounds like these ones that normally act as defenses for generalist insects because normally they are bitter, they can actually act as susceptibility factors like oviposition cues in specialist feeders. Okay, so besides the chemical information provided, our results also may have implications for agriculture or forestry. For example, the use of resistant plant varieties may be an important strategy to sustain wood and biomass production in willow tree plantations. Yes, since selected PJs correlate with sawfly damage, these defensive compounds could be used as early detection of susceptibility. For example, you can take leaves and simply dip them in methanol and analyze them, or you can also select genotypes against to increase resistance. Okay, as you know, also after herbivore damage, the chemical profile of the plants change, and these changes in the phytochemistry can affect the future herbivore selection for feeding, for oviposition, and also they can affect the development of the next generation. So our last objective, I will show you, is to study if plant chemistry changes after willow damage. So, and I will show you why we studied oviposition and which are the effects of egg deposition. Remember that this is how she laid the egg, and she used to make a small injure to the leaf. And also because herbivory normally starts with an egg, so an egg is like a signal for the plant. An egg predicts a new insect generation. Plants recognize eggs and display defense mechanisms, and eggs can also induce priming. In another systems, yes, we don't know here. So our question was, do volatiles change after oviposition as posterior feeding? And does it affect the oviposition preference of the female? Well, this is the methodology. It's a little bit complicated, maybe. We started with two groups of plants, one group without eggs and the other with eggs. And then we will call this day -5 because day 0 will be the day in which neonates hatch. So we have these two groups, and then after hatching, we will leave the larvae here to develop, and here we transfer larvae. Yes, Camila did it because she has very good hands for that. So Camila transferred neonates in this plant. So now we have 4 groups: without eggs and feeding, with eggs and feeding. And then we continue the development of the larvae. And we measure volatiles at day -1, so 4 days of egg development, and also at 4 days of feeding damage. And we measure here oviposition preference. This is how we collect volatiles by headbase. And these are the profiles of some of the sample without eggs, with eggs, and you can see that only the presence of the egg, already you can see, for example, a small peak of ossimen here and a small peak of farnesan. And also when you add the feeding damage, normally you have more volatiles. For example, here you can see the huge peak of ossimen after larval feeding. These are all the compounds we found, and these are the significance by feeding or egg deposition. But in order to visualize it better, Camila did a correspondence analysis among treatments, and you can see that the odor blend for the plant without eggs, so for the undamaged plant, was different from the other blend of the plant with the egg, in which I brought this from the table. Ossimen and farnesan appear like small peaks, and then after damage, well, you have like the blends differentiate more, but don't differentiate among them. So the volatile profile differ depending on the type of damage. Specific volatiles were emitted after only oviposition, and we didn't find evidence of priming. So because here these two treatments were pretty much the same. So the question now is, does it affect the oviposition preference? Well, this was a very simple biocide performed by Selina. She offered one ovipositing female, one lives with eggs and lives without eggs in a cage. And this is what she found. This is the number of eggs laid, and there were no differences. So for the female, the egg presence didn't affect oviposition preference. Okay, so now, oviposition per se stimulated jasmonic acid production. I haven't shown these results, and also specific volatiles emission. It did not affect females' oviposition preference. Actually, we don't know about natural enemies, but this is encouraging, but because we know that ossimen and farnesan in other systems attract egg parasitoids. And after feeding damage, volatiles were strongly stimulated. So, you know that it was proposed that phytochemical variation in tree tops has the potential to shape insect community diversity and population strategies. We think that plant resistance in willows may act both directly on herbivores, as I showed you before, or indirectly maybe through this volatile emission to attract natural enemies. And this might be important because it might have implications for willow breeding in the search for more resistant varieties. Well, this is just to tell you something about the future directions. We are starting to work with Salix humboldtiana. This is the only native willow in South America, and well, we have some populations which are not very domesticated, so we are trying to compare these non-domesticated or with little domestication populations with the high domesticated commercial genotypes, because we think that it might be a genetic resource important in the context of global climate change. And we are doing this with Camila, and also the first works were performed by Gabriela Valladares, postdoc. And then we also started to look underground because we know that mycorrhiza can affect also the phytochemistry, and it might affect so the interaction with insects. And we are starting to do that with Paula Galota. She's also a PhD student, and with the help also of Marino Massini. Okay, so thanks a lot. Muchas gracias to the girls that did a lot of work with in her PhD thesis, Celina and Camila, also Jorge Zavala and Andrea Vega from the University of Buenos Aires, Romina Barroso, Maria Victoria Colaraos, and also Peter Thiel from USDA, Teresa Cerrillo, who did a lot of these surveys, field surveys, and also for the phenolic glycoside, we had a lot of help of Jonathan Gershenson and Michael Reichel from MPI in Vienna. Thank you for your attention.
Okay, so it's time for questions. If anybody would like to ask anything to Pato, please let me know and I will unmute you. Or if you would like to type your question, please do so.
Was I too fast, maybe?
I don't know. Like,
it's my I always talk very fast.
Well, I was going to ask about the what would be the next steps, but you kind of like already mentioned that. Have you been able to start those new directions or those future directions, or is it something that's going to start in sometime?
We started, well, actually with Victoria Colaragos, she's there, and also with Gabriela Valladares and Teresa, the girls from Tucumán. We started, good speed. We started to see these volatiles in Salix humboldtiana, and so the very first, oh, Tyler, thank you. The very first results show that there might be nice defenses in that tree, but then we really need to continue a little bit because what was our first paper actually showing that maybe we have good defenses, but we really need to continue. So that is why now Camila Davida, she's comparing one of our commercial trees with this ones to see if there are some differences. We didn't find much in the behavior of the specialized insects, and so we are in the way. With the mycorrhizae, we're just at the very beginning.
Oh, okay. Thank you so much, Pato.
You're welcome.
Any other questions? So Tyler asks if you could discuss more about the eggplant interface and what might be going on there to induce volatiles.
Well, actually, I did a big job with that because you know that you have the plant. I didn't bring the scheme today, but you have the plant and you have the epicuticular waxes, and it is sometimes difficult to think that you might have these polar compounds in the epicuticular waxes because they are very non-polar. So we did a very, very detailed work, And we found actually some, especially I think tremulacin in the epicuticular boxes that they may leak from inside, yes, because they are polar. So that is why it was very difficult. I try a lot to do like behavioral experiments by adding phenolic glycosides to the leaf and try to see if they were like very good oviposition cues. This is very difficult to do. It doesn't work because this, as I show you the picture, the video, they are very picky. Yes, they will go look around a lot till they found the right place to lay the egg. So I couldn't do manipulative experiments with these cues, but I really think that phenolic glycosides are part of a fingerprint. I'm not sure if that was the question.
And then we have another question from Hector. Oh, sorry. Asking if intercropping would affect these interactions and if willows are planted in monocultures or polycultures.
Yes, actually, that is one of the ideas where I work. Willows are planted in monocultures, and mostly from one clone. There are some other clones, and actually there are a lot of clones available. So we are trying to tell them to do this intercropping because I think that they can disrupt a little bit the behavior of the oviposition behavior. And I think that without oviposition, you will have no damage because the adults don't feed.
Okay, so I guess those are all the questions. And thank you so much, Beto.
Thank you.
So now we'll be jumping straight to Tyler. So Tyler has been experiencing some tech difficulties. So we'll see how that goes. Let's see. And Tyler?
Hey there. Everybody able to hear me?
Yeah, perfect.
Well, that is great. I'm going to try sharing my screen here. Patricia, thank you very much for that. I think you and I should talk more about that egg plant interface. I've got a plant trait that might be working at that very level. All right, are you guys able to see my screen?
Yeah, we can.
Okay, great. So I'm going to apologize for the technical difficulties. I don't know what was going on. And I'm going to also apologize for any beeping that you might hear in the background because they are clearing the massive amounts of snow off of our street that we got not too long ago. So I'm going to tell you about CAM, the Cereal Aphid Manager app. Now, this is an integrated pest management tool that brings together things like economic thresholds and how to scout and puts it all into one place. So it's kind of a long story, and I'm going to take you back to 2012. And first, though, I'll acknowledge my co-authors. So Earl and I worked on CAM Part 2, let's say, or Dynamic Action Threshold Part 2. Christelle Olivier and Owen Olford put in the original proposal back in 2012. And John Gavlowski was the innovator of this idea and wound up being the man who got the funding going. And he was also involved in doing the Manitoba sampling. And then Elam Karimi was our wizard in Ottawa who actually put the app together for us. So if hopefully I'm coming through all right, here's my email address down here at the bottom left, and this is my Twitter handle right there. So both ways that you can get a hold of me. So the inspiration for this whole project actually came out of a University of Guelph project called Aphid Advisor that was run by Rebecca Hallett. So she actually had a PhD student named Christy Belay, who is now a professor down in the US, and they used a dynamic action threshold to predict the effects of the beneficial insects on the field on soybean aphids. So they put it into a smartphone app, which is a smart idea. And as you can see here, we've got an iPhone and a BlackBerry. So that kind of tells you the age of Aphid Advisor. And So it is a pest management decision-making tool. And so it basically tells you to spray or not to spray when you're looking at soybean aphid. And so they did some follow-up, and they said that using the app wound up reducing pesticide applications by 100%. So it made the grower 5 times decide not to spray his field for soybean aphids. So what is the problem with aphids? Well, we're all entomologists here. We know that most aphids are just, oops, let's go backwards. Yeah, sorry about that. Previous. There we go. Aphids are all female, and they're basically born pregnant. So here's a cereal aphid. This is an English grain aphid, and we've got 7 babies ready to go inside. So they're born live, and they start feeding right away. So this leads to rapid population increases for aphids when you don't have to worry about mating. You don't have to have half your population as males. Everybody comes out female, they just start feeding, and the population goes up and up and up unless something acts on it. So here's another mama cereal aphid here, and she's got 3 aphids on this side, and she's got 4 aphids on the other side. So it looked like her reproductive output was about 7 aphids per day there. So we could have anywhere from 2 to 4 to 7. It all depends on the species and a few other things as well. So here's that increase of the aphids over time unless they're acted upon. So 22nd of July, no aphids. Came back a little over a week later, and we're already at 7 aphids per head. So when you're scouting cereal aphids, you're actually counting aphids on the tiller. And most of them are going to be born up in the head. And so blue line represents number of aphids. About a week later, we hit 16 aphids per head, which is over the economic threshold. So this is a point where damage is going to start happening. But what we found was that we had 13 spotted lady beetles that moved into the field at this point here, and the population actually dropped down to about 5 aphids per head less than a week later. So another thing that could be happening here is that what you have going on in mid-August is senescence of the crop. And so this is something that I always needed to think about too. So what stage is your crop at? And so we actually worked that into the dynamic action threshold too. And at this point up here, we were at the medium milk stage in the cereals. And so those grains are still filling. So they're still building up starch. And we can still be taking damage at this point. But once that grain hardens and we're at the soft dough stage, aphids don't do any damage anymore. So the app and the dynamic action threshold take that all into account. So we see this tiny little bit down here at the bottom. So these are aphid mummies that started to show up. So we really didn't see much in the way of aphid mummies until we looked at the population after it had started to crash here. So maybe they had a bit of an effect on the population as well, but it was probably these 13 spotted lady beetles that moved in. So the week before we found 1 just in our looking, and up here we found more of them with a sweep net. So the project conception came out of John Gavlowski going to the Pest Management Center's priority setting in 2012, and he said, you know what? There's this cool dynamic action threshold, and it works for soybean aphid. Wouldn't it be great if we got it working for cereal aphids? And so then the Pest Management Center, and so they're a branch of Ag Canada, and they work out of Ottawa. They put together some money, and Christelle and Owen applied for it and got a small budget, 2-year project, about $80,000, which paid for summer students and eventually paid my salary too. So this came about at the tail end of my PhD, and I blew through my funding in the first 5 years, and then had a year or so of no funding. And so I supplemented my zero income from my PhD with working on cereal aphids in this project. And so the idea behind this one was to develop and validate this dynamic action threshold. And so we needed to know what was out there, and we needed to know what effect they were having on the populations. And so when Christelle, there she is right there. I don't have a picture of Owen to show you, but I'm sure you can find him online if you like. So Christelle wrote up the project and then did the sampling in 2012 and tried to figure out this dynamic action threshold. So she brought me in and away we went, and I started identifying insects that they had collected in 2012. So that was step one. We needed to survey what was out there. And step two, then sample these populations and then try to compare what was going on with the aphids in the field to the predictions that any dynamic action threshold would make. So I had to take that dynamic action threshold and adapt it from soybean into cereals. And so what am I talking about when I'm talking about a dynamic action threshold? So the action threshold is where you need to treat your insect problem before it becomes an economic problem. So that's our economic threshold. So the action threshold and the conventional action threshold, we can call it the CAT, happens before the economic threshold. So you spray your aphids here to keep them from getting to this point over here. But it doesn't, the CAT doesn't take into account that controlling pressure of natural enemies, whereas the dynamic action threshold changes based on the number of natural enemies that you've got. And so most of the time the recommendations are just look around for natural enemies and maybe some parasitic wasp mummies, but there's no real quantification or measure of their impact on the population. And so what I was trying to do was to quantify their impact on the population. And so we do that using something called a natural enemy unit. So this came from the soybean aphid project, but it also came out of the literature before that as well. So a natural enemy unit then takes into account this value N. And so this is the number of whatever natural enemy you've got, and then V, which is an important one. So V is the voracity, or how many aphids are killed or eaten per day. So you put those together and it comes in as one value called a natural enemy unit. So in 2012, 2013, the sample was here. This is Saskatchewan. It doesn't typically look like this on a map. There's not wheat everywhere. Our bread basket where we grow wheat is about right here where my pointer is going, down into the bottom of Manitoba here. And then the top part is all Canadian Shield. So it's all rock and trees. But Saskatchewan is a really big area for wheat growing. So we needed to know what aphid species we had. We needed to know what natural enemies were out there. So we surveyed the fields, a few in Manitoba and a bunch in Saskatchewan. Here's what the fields look like in the springtime. So not a whole lot going on. And sometimes we get road washes out. So here's my sampling team from 2012 and 2013 standing in a sunken hole. So we were looking at the aphids and the natural enemies on single plants, but also with a sweep net. That's me sweeping a wheat field right there. And so we had some abundant aphid years. So you can see some here on wheat. So we looked at wheat, we looked at oats, and we looked at barley as well as our three main cereal crops that had good economic thresholds for them. So you can see the aphids building up here. These are probably going to be a bit of a problem. And what we're looking at are the English grain aphids here. And of course, your seeds are filling right here, and your seeds are filling right here, and the aphids like to pick on that area where all of the sugars are flowing into the seed. So what we found was basically two different species of aphid out there. We've got the bird cherry oat aphid here, and then the English grain aphid. You can see our English grain aphid comes in a few different colors. So we got a bright green, we got kind of this dull orangey color, which is sort of green still, and then this bright red. And so we actually started looking at the two different morphs in 2015. So they were not red in 2013, 2014, but in 2015, the English grain aphids started to present as red. So here's what we got for bird cherry oats, and this was pretty similar as we went from year to year to year. We had the two different aphid species and not a whole lot else in there. So interestingly, we discovered that the green morph and the red morph, we can actually turn a population of green morph into red morph by increasing the light intensity. And so we published that just this year. You can take that red morph and turn them into a green morph by decreasing the light intensity. So that was really interesting. And Kevin actually documented in the field here that on the top of the wheat heads, we had more red morph than we did on the bottom, where we had more green morph. And so it is possible that we've got some kind of a light effect going on the tops of the wheat heads versus the bottom of the wheat heads. So out in the field, our main predator was the 7-spotted lady beetle, and we needed to know what the voracity was. So there's been a lot of studies that tell you how many aphids can be eaten a day by all these different lady beetle species. And so from the literature, then I could get the voracity. So we got 48 aphids per day for an adult and 72 aphids per day for a larva. So these numbers go into that natural enemy units equation. We had 13 spotted lady beetles as well, which is a native lady beetle. Now, this one's interesting, the 7-spotted, because it's actually introduced to North America. And there was a similar project to ours that was looking for the Russian wheat aphid in Saskatchewan, back when the Russian wheat aphid was invading North America. And what they found was they found about 13 different species of lady beetle in their wheat fields. And this was back in the 1980s. And I've got their report, and one little throwaway line in their report said, and we started finding a few of these 7-spotted lady beetle, and we're not really sure what effect they're going to have on the overall population. Interestingly, now about all we find in our wheat fields in Saskatchewan are the 7-spotted lady beetles, and those 13 other lady beetle species, few and far between or not present at all. So just an interesting aside there. 13-spotted lady beetle, a little bit smaller than 7-spotted, and they eat less. So these two made it into the dynamic action threshold. Our green lacewing larvae, voracious predators. We didn't find as many of them out there, but they'll eat about 30 aphids per day, and doesn't really depend which species we've got, but we do have Carnea and Oculata out in the field, and you can tell them apart by looking at their head capsule and these patterns on it. And they're a great predator. They're a piercing, sucking one, and they leave behind the skin of an aphid after they finish drinking the aphid's insides. So Rebecca Hallett ran into a problem. Her and Christy Belay had to figure out how many aphids per day the minute pirate bugs would kill, because there wasn't any information on that. So they actually found out that nymphs killed 8 per day, and the adults would kill 11 aphids per day. So not a top predator, but these are minute, like the name suggests. They're very small, and about the same size as an aphid. So it's actually pretty impressive that they can take out 11 aphids in a day. So out east, where Rebecca did her study, they have Aureus insidiosus. Out west, we've got Aureus tristicolor, and the difference is just coloration of the clavus. So I had to use the Aureus insidiosus numbers because we didn't know what tristicolor might do. So just thinking that they're about the same. We had a few damsel bugs out there, and they'll take out about 19 aphids per day, so that's their voracity there. And then we found that we basically had just one parasitoid, Aphidius avinaphus, and it dominated the parasitoid population. If you know anything about aphid parasitoids, this wasp comes along and stings an aphid, and then about 12 days later, you get this aphid mummy, or 10 to 12 days later. And so the aphid is dead, and inside is one single wasp larva. And then it pupates, and out comes a female wasp, or a male wasp. And those were two of the things that we needed to figure out. So we had this huge hyperparasitism rate as well, where we got these wasps that would come out. And I'll talk more about those and the male to female ratio later on as well. And so there wasn't any information on the voracity of Aphidius avinaphus anywhere in the literature. So just like Rebecca Hallett did with the minute pirate bugs, we needed to figure out how many that would kill. And so I was using Aphidius colemanni as a proxy. And so you get a natural enemy unit of 0.6 for one Aphidius colemanni female. So who knows if that was about the same, and we needed to figure it out. But first, I had to build this stat equation. So I'm going to bore you to tears here with some math. So NT, this is the predicted aphid population. T, of course, represents time. And so it's this number that gets compared against the economic threshold when you do a prediction of the growth of the aphids. So this is your initial aphid. So this is your mean number of aphids that you get when you count aphids on the cereal heads. This is a constant, this E. And R represents the intrinsic rate of increase of the aphids. And so why is R important? Well, this tells you how fast the aphid population increases. Now, we're all entomologists here, and I know that you're probably yelling out at me, well, doesn't your rate of increase for aphids change based on degree days? So if it's hotter, shouldn't they reproduce faster and reach their adult form faster? Yes, you guys are very smart, and you're right, they do that. Now, I was trying to figure out, okay, how do I get a prediction for 7 days in the future? I don't trust the weather, man. I don't trust anything that they're saying for 7 days in the future. How am I going to get that into the equation? Do I need to connect the dynamic action threshold to some kind of online server that tells me what the forecast might be? And it was all very confusing, and I didn't really see it working properly. So what I did was I went to the 3-week time point that we usually get aphids in Saskatchewan and looked at the average temperature over the last 20 years. And the average temperature was pretty much just 20 degrees Celsius. So if you consider daytime temperature and nighttime temperature, it just averages out to 20 degrees Celsius. So for simplicity's sake, I just used the reproductive rate of English grain aphids at 20 degrees Celsius. Got it from Ackerman and Dixon, 1989. And yeah, so then the natural enemy unit equation, I talked about that, number of your natural enemy, and then how many aphids it kills per day, and then you sum it all up for all of the insects that are killing aphids in the field, and then you get a natural enemy unit total, which goes into this complicated, but not actually that complicated, equation. So this is the number that's going to compare. This is your predicted number of aphids, and this is the sum of everything that's going on in the field. So this is your rate of increase of aphids. This is the number that goes in. And you might notice that these two terms are almost the same, but not quite. So our rate of increase is here on this one, and it is outside on the other one. And so this takes into account if the aphids get killed before they reproduce or if the aphids get killed after they reproduce, and then divides it by 2, because you don't actually know what's going on in the field. So this equation represents a lot of educated guesses smashed into one final equation. And yeah, then it goes into the aphid calculator, and it looks very confusing, and it probably would be if you tried using it like this out in the field. And so we thought, well, we better compare it to what's actually going on. So a red line, this is the actual aphids, and the black line is the predicted number of aphids that the DAT uses. So under a situation where we don't have a whole lot of predators, we just had a few 13-spotted lady beetles in here, it seems to predict really well. And so the lines are matching up fairly well. So we had a few fields where the lines matched up really well, and a few fields where they just didn't match up very well at all. And then I thought it would be fun to show an exponential growth of the aphids over the same period of time. With our start of 6, we get over 41,000 aphids if nothing affects the population. So obviously something's slowing down the population in here. So we did this just to make sure that that DAT was working. And we finished up that project and said, hey, wouldn't it be cool if we put this into a smartphone app so that people had it in the palm of their hand and they could use it to scout aphids? And so in 2015, it was good it wasn't 2014 because we actually didn't have any aphids in 2014. We got funding from the same group again to turn this dynamic action threshold into a smartphone app. So a bit bigger of a budget over 3 years. And Earl Svensson and I, he's our knowledge and technology transfer guy, we went into the project trying to refine that dynamic action threshold and turn it into a smartphone app that we would just release for free to anyone who wanted it. So our goals were to teach people how to scout for cereal aphids and teach them what the beneficial insects look like and what they could be doing in their field. So I put this first because this is my primary goal, is to teach people about them. And then it gives you a scouting calculator as well, keeps track of what's going on in your field so you can use it from 1 year or 1 week to the next and know what happened last week. And then decision support tool is what it actually does. So it makes recommendations that you can follow or not follow. And one of the ulterior motives was to actually protect the beneficial insects that were in the field, potentially reduce insecticide inputs. So we use the economic thresholds from that we typically use in Saskatchewan. So it's around 15 aphids per tiller prior to this soft dough stage. And that's very important. If it goes after the soft dough stage, you don't need to get into your field and do this W pattern here. So this is what's typically recommended for scouting. And you take this W pattern and you look at 5 different tillers in 5 different places. So it gives you 25 serial tillers. So you've looked at the heads, you looked at the leaf, and all the way down, and you added that all up, and you get a mean number of aphids. Now, part of what we needed to do was to refine it based on these parasitoids, and we didn't have any data on Aphidius avinaphis, so we had to figure it out. And this was actually a really complicated experiment to do. So we emerged some Aphidius avinaphis from the field in 2015, and then we tried to see how many mummies they would produce in one day to get that voracity number. So here's what we wound up doing. So we challenged them with English grain aphids, that represents this blue line here, and we get an average of about 31 aphids per day that got mummified by the female Aphidius avinaphis, which is about half of what we predicted based on what Aphidius colmani seemed to do to aphids. So right away there, our veracity from that first project was too high for parasitoids. What was really interesting was we also challenged them with the other aphid that we find in the field, the bird cherry oat aphid, and we basically got no mummification of the bird cherry oat aphid. And so after 2 days of challenging them, we actually ran out of aphids. So this is, you wouldn't think that you would ever run out of aphids, but when we were putting over 1,000 aphids per day being challenged by these parasitic wasps, we ran out of aphids pretty quickly. So we had enough English grain aphids to continue up to about day 6, and that's about how long one of the wasps lasted. So you can see they sort of started dying off around day 4. So the wasps last, say, 1 to 5 days, and they were taking out 31 English grain aphids, but not bird cherry oat aphids per day. So we're not really sure why that we weren't getting mummification of the bird cherry oat aphids, but here's our natural enemy units, and so these guys will work for English grain aphids, but maybe not as a biocontrol for the bird cherry oat aphid. And we were able to see that they were stinging the bird cherry oat aphids, but maybe the bird cherry oat aphid are able to encapsulate the eggs of the parasitic wasp and keep them from developing inside their body. So that's really interesting and something for a future project. Now, we needed these wasps to kill enough aphids so that we could get that natural enemy unit of 0.31. Now, this is a picture of 16 mummies, and each of these mummies then, can it be a proxy for one wasp? It could, but we need to refine it a little bit more. So in this case, we've got 16 mummies, and if I look at it and say we got 0.31 as our natural enemy units, we get a number of 496 aphids that this amount of mummies is going to take out of the equation. And so is that valid or not? Well, if you remember back to our hyperparasitoids, we had a lot of these beautiful looking Asaphis suspensus, they're a pteromalid, and they were taking out 60% of the primary parasitoids. Dendroceras bicolor, they were doing some effect as well too. Here's the males, you can see the difference here in the antennal segments. Very interesting. And then some Alozysta species, but collectively we were losing 60% of these wasps to hyperparasitism. So we had to work that in, and then that reduced the number down to about 12 aphids per day. Then you have to think, okay, well, not everything that's coming out is going to be a female and able to sting and keep attacking. So sometimes you're going to get males. So then I worked in the ratio of females to males, which is 6 to 4, and then our veracity gets down to about 7 aphids per day. So that is a whole lot different than the original starting number. Working it into this, we had to look at it as well and kind of compare them again. So like I did before, here's our English grain aphid, and this predicted DAT is way higher than what actually happened in the field. So exact same field, for the bird cherry oat aphid, when I split them apart, it matches nicely. And so the predicted and the actual are matching up nicely together. And so we're still not getting a great prediction for what's going on with English grain aphid in this field, at least. So we still had the task of putting it into an app, and away we went. And we actually, so I'm going to not bore you with all of the government details and all of the brick walls that we ran into making the app, but we got it done, and we actually released it right at the project end. And what you see on the screen are just screenshots from it. So when you're using it, you go into a field assessment, and you can do as many fields as you want, and it will just keep track there. So if you come back to my wheat field here at the Saskatoon farm and you click on that, you can see what you did last week and the week before and this week as well. So it stores all of your sampling in memory. This is important when you're starting out. You tell it what stage most of your crop is at, and if it hits early dough, you're going to get a message that, hey, you don't even bother needing to do your field. But of course, I like scouting aphids, so I just ignore that, and you can keep going even if it is past the scouting stage. So, got the warning here, hey, don't worry about scouting, but I did anyway. So you take that W pattern, and at each point here, you're going to get a pop-up, and it's called an observation point. So you got 5 observation points. Each of those represents 1 tiller that you're going to be counting. So 5 at each spot winds up being 25. And you get to enter the number of each aphid species that you see there. So if you're like, well, I don't really know what a English grain aphid looks like, you click on that English grain aphid and it blows it up and says, okay, you're looking for long black cornicles, you're looking for black patches here, here, and here. It gives you a body color variation, so you kind of can say, I think I'm pretty confident that that is an English grain aphid that I'm looking at. And then at that same point, you count the number of natural enemies that you see on those 5 heads and also in the area around you. And this is another one of those estimates that we're still kind of working on based on using the app. So we've got all the ones that we found over the project years, and we've got them in there. So you can get a prediction after 25 heads, and it always tells you to sample another because I don't want anyone making a decision based on 25. So after 50 tillers, it will give you what it actually thinks you should do, and then you can decide based on, if you really don't like aphids, you can take your 5 aphid per tiller recommendation and all the way up to the 15 aphid per tiller recommendation. So you'll actually get 4 different recommendations based on how comfortable you are with having aphids in your field. And then it'll break it down by species for you, tell you what the natural enemies are. And in the newest release, it'll give you the average here, so you don't have to calculate it yourself. And it'll give you a nice little graph based on what you looked at last week and what you looked at this week. So it has the look and feel of a government app. This is the main screen that you'll see. And this is the kind of button that you'll see on your cell phone. And with the touch of a button, it all becomes French because we're the government and we have 2 official languages in Canada. So we made the app twice, once in English and once in French. And if you're so inclined, you can point your smartphone QR reader at the screen, and this will take you to either the Apple Store. Okay, I'll go back. It'll take you to the Apple Store, or it will take you to the Google Play Store, and you'll be able to download it just like that. So the app is free from the Queen of England and the Government of Canada to you. So here's my acknowledgements, everybody who helped out on the project, especially Ellen Karimi and her team in Ottawa who built it all. And I'm going to open this up for questions and stop talking. And there's all of my contact information, and check out at Field Heroes as well to find more information on all those beneficial insects that I was talking about. So I'm going to stop sharing.
Okay, so thank you so much, Tyler. And yeah, we'll start with questions for Tyler, and we also have one more question for Patricia. But we'll start with Tyler.
Are you listening to me?
Yeah, we are.
There, we can hear you, Eduardo.
I have been struggling to unmute me. Thanks, Tyler, for your very interesting talk. I have about 5 questions. I don't want to monopolize, but I was just wondering whether you were thinking about the possibility of turning all the work you have done in quantifying the effects of different natural enemies in putting value, economic value, to the biological control service, ecosystem service that they are providing by making the difference between what is the situation without or with or without the predators or the natural animals in general.
Yeah, that is a great idea. So, yeah, we need to figure out the value of each of those to wheat that year and mix in to the equation things like how much would you spend spraying if you were to spray.
Exactly. Yeah. I did that. I tried to do that with parasitoids of stem borer in Argentina, and it was just an theoretical exercise, but I think it's worthwhile trying it with some good data.
Eduardo, did you get my email address there? Would you mind sending me that paper? I'd love to read it.
It didn't come out. It was just an exercise, and it dried out. It didn't follow it.
Ah, okay. Well, I can speak to
just
you.
An idea.
So we've got wheat midge up here. Do you guys deal with wheat midge down where you are?
I don't think so. I don't.
That's good.
Doesn't ring the bell to me.
Okay, you don't want it. So that's a bad one for wheat. Now, they actually quantified the effect of the main parasitoid of wheat midge, and so I would probably base anything that I did from cereal aphids off of that. And so they actually got, it was millions of dollars over the outbreak years. Like the economic benefit from just having this one parasitoid was millions of dollars every year in reductions in pesticides. Yeah, that's great. All right. Eduardo, did you want to ask 4 more questions?
May I?
Sure.
Sure. We also have another question from Patricia, but I'll read that when you're done.
Okay. Yeah, I see it there.
So I can wait.
No, Eduardo, you go ahead.
Okay. I was wondering whether do you think is it possible to work with a functional response? Because I mean, calculating the voracity of different natural enemies, it would be different whether you have a low or high aphid density.
Certainly, yeah. Yeah, and we actually don't get much in the way of natural enemies until we do have a high aphid density.
Okay.
So one of the things I'm struggling with is something like the searching ability. So you need to take into account the time of searching. So if you're a ladybug larva, you're not flying around from head to head. You have to walk from head to head. And so taking into account the amount of time, your voracity might not actually be 72 aphids per day like you would get if you were in a petri dish with 100 aphids, right? You're basically, you're up to the buffet and you just eat as much as you can. But when you're out there and you have to search for your dinner, that's going to be different. So I could probably just continue working on this for the rest of my career, I would think.
Okay. And the last one, if I may, is just I leave it there. Have you considered the possibility of developing a sequential decision protocol, including the natural enemies? That would be an interesting, maybe too subtle for decision-making application, but for the sake of research, I mean, maybe it would be interesting to develop this sequential decision sampling protocols. Yeah.
So I'm not sure I'm 100% understanding what you mean about the sequential sampling. Do you mean
Instead of setting a fixed number of sampling units, you decide when to stop your scouting based on the density of the pest, and in this case, based on the density of the natural enemies.
Yes, no, I see what you're meaning now. Yeah. So in the app itself, you can go up to 4 different samples per field. So you can get 100 heads per field. So you can actually sequentially sample. And my idea with the app was actually to continue to use it and get that sequential sampling data from different fields. Now, our trouble is trying to get to enough fields where things are going on with aphids and natural enemies. But over time, we can do that. So I'm actually kind of building a data set using the Serial Aphid Manager app to answer those sequential type questions. Another one, too, we can look at is maybe you don't need to spend a whole lot of time counting the aphids, but if you just put in like binomial sampling, and I know they did that in North Dakota, right? So you count, if you get up to 19 heads with aphids, you're at your economic threshold. And so the app is designed to save that kind of information so you know how many heads had aphids. And then hopefully in the future, after we've used the app enough, then I can actually answer those questions and say, okay, so in Saskatchewan, we can use this sampling as well. And it works out. So one of the things I wanted to do with the app was to let the growers have the opportunity to send me their data so that the app became a sampling tool that people could it became a sampling tool for me to just keep getting data. And so I wanted it to be done automatically. And then Earl said, whoa. Growers don't really like the government getting data from them automatically. And so we now have a way so it doesn't get automatically. So it was like, the growers don't want Big Brother watching them. That was one of the thoughts he had, and I said, okay, that's fine. So there's an opportunity, though. So once you're done your sampling, you can hit, and this is the second iteration of CAM, you can actually hit a button and you can email it to whoever you want. So you can email that data to me if you want. So if you're going out in a wheat field and you take Serial Aphid Manager with you, you could do all of this and email it to me, and you could help me increase my data set and the work on it. So there's my challenge to you.
Okay, thank you very much, Tyler.
Thank you for the good questions. So how about I answer Patricia's? And thank you very much for saying it was a great talk.
Sure. Let me read it out loud. So Patricia says, since it looks like natural enemies do a good job, do you get best results in a more diverse landscape? Would you recommend planting some biodiversity around crops?
That is an amazing landscape ecology question that we get quite a bit, actually. And it's always the scientists that ask that question. I'm sure we might get better results in a diverse landscape. But if you know anything about Saskatchewan fields, they are huge. So we're talking like a quarter section, could be maybe 1 or 2 fields. So the diversity in our landscape is not great. So maybe we might get ecosystem services from things like a natural enemy, or a natural area, but those natural areas are going to be really far from the center of most of our wheat fields. So it's a great question, and it's something that I would like to look at. And there are people in Canada who are looking at the effects of these natural areas on things like yield and the insects that are in canola. But it's going to be probably not difficult, but it'll take a concerted effort to do that in Canada, I think. So great ideas, and I don't have a great answer for you on it.
I think Patricia has another question. So
yes, no, thank you, Tyler. I asked you that because we were working with ants, with the leaf cutting ants, and also in this forest, in this willow forest, and we found that the threshold, yes, the economic threshold or the action threshold, I'm not really sure which one, is depending on the landscape, you will have different thresholds. And actually, I thought that maybe it's a very good idea if you can borrow some natural enemies from some diversity, and you can manage the landscape to bring this kind of natural enemies. We found that with the ants, and I think that it should work really well.
Yeah, so your ants were the natural enemy in your system then?
Well, actually, we didn't measure natural enemies, we measure damage. And the ant damage diminishes when you have biodiversity around.
Oh, yeah. No, that is very interesting. And my guess is that biodiversity would have a really good effect on the seven-spotted lady beetle because they overwinter as adults. But we're also looking at things like temperature and snow cover as well. Because if you're overwintering as an adult and you like sort of the natural areas, you need a good blanket of snow to keep you alive during the winter in Saskatchewan. So we actually had a really low population of lady beetles this year. And of course, with COVID, we didn't have a great chance to study what that could have been caused by. But things like snow cover in the natural areas, I think in Saskatchewan, we're going to have a huge effect, but also maybe the flowering plants as alternate food source for our parasitoids. But I'm not going to hang my hat on the parasitoids as being able to control populations up here of aphids. It seems to be more the generalist predators that are doing the better job.
And it happened to me the same, that farmers don't like much biodiversity around. Sometimes it's difficult to manage, so they don't really like it that much.
Exactly. Yeah. So, Eduardo says we could put a value on the border vegetation, and that's a great plan. So we're also looking at when we go into the fields, we also look at what's in the ditches, and so our ditches are a mix of flowering plants and brome grass and alfalfa, which does flower too. And I think those are potentially a lot of the refuges for the natural enemies that are coming into the fields. But also we've got little copses of trees that happen and little bodies of water as well. And so, there's a lot to do there. Alejandro at University of Manitoba, he studies effects of landscape ecology on aphids, and Hector is mentioning Arash's paper, and so Arash was working with Hector and was working with Alejandro at the University of Manitoba, and they did find landscape effects for cereal leaf beetle biocontrol. So the cereal leaf beetle is basically controlled by one parasitoid that Hector's group there at Lethbridge wound up bringing in and releasing. And so they did find landscape effects there. So thank you for that addition, Hector.
Thank you.
It is possible. So along with Alejandro, we're looking, and Hector, we're actually looking for landscape effects on flea beetles and potentially on their predators and parasitoids as well, if we can find them. So we're trying to answer these questions, and hopefully we'll get there and have a better idea.
Okay, so Tyler, I have two more questions for you, and then one question for Patricia. So Tyler, Dylan asked, or he says, I miss which of the two parasitoids was actually successful in parasitizing the aphids, but I was wondering if you know if any of the parasitoid venoms has been identified.
A good question. So we have Aphidius avinaphis, and that was the primary parasitoid that we found most often in the cereal aphid system. Now, when you say venom, are you talking about maybe the way that they overcome the host's immune system, Dylan?
He's using the chat box in YouTube, so I hope he's actually listening.
I can't see what he's saying.
Yeah.
Right.
but I assume so.
So-
And, yeah, no, you go. Sorry.
Yeah, well, what I was wondering about with the two different aphid species, and one being stung by the parasitoid but not developing into a mummy, was some kind of a host immune response. So it might not, maybe it will come down to a difference in venoms, or maybe it'll come down to something else that because not only do eggs get injected depending on the parasitoid, but they can also inject viruses that will help overcome the host's immune system and prevent that encapsulation of the eggs. So there's a whole system that I could look at there. Like, we should be able to open up the aphids and look for encapsulation of the eggs. It would be a great little side project. If you're interested, Dylan, I know where you hang out.
So that's awesome. And now a question for Patricia. So Eduardo asked, what about spatial scale of preference at the field level?
Yes, Eduardo. I haven't done much about that, but I just have a meeting, I think it was this week, and they some people, they told me that they have data about how the sawfly is moving in different depending. So the idea is to see if the sawfly is moving in depending on the willow species and the scale, but I don't really did that measurements. I think there's something, but we have to check it.
Okay, so thank you. And there's a follow-up question for Tyler. So Dylan says, yes, essentially how their oviposition fluid can influence their host immune response.
Excellent. Yeah, I would love to follow that up. It sounds like a good master's project, potentially. Yeah.
And then I have one.
I'm not going to say our.
Oh, no, you go ahead,
yeah.
Tyler.
No, I was just going to say
with
our.
So.
Yeah, no, I'm not going to say it.
I have a question.
I'm going to say.
Oh, okay. Yeah, you go with your idea, and then I ask.
I was going to say, Dylan, we have a population of bird cherry oat aphid, and in August we can get some wasps, and so we can give it a shot. So how about I get in touch with you in August, and you can make that a chapter of your thesis.
I'm pretty sure that Dylan will be super excited about that. And then I have a question myself for you, Tyler.
Sure.
That switch between one morph and another one for the aphid populations, is that something that happens throughout the entire life cycle of the aphid, or is it only present or only achievable at some point in time?
I lost what you said there. Can you repeat the beginning of your question?
Yes.
Kind of fuzzed out on.
Yeah, sorry. If that change between the green morph and the red morph is something that happens at any point during the life cycle, or something specific?
No, that is a good question. So where does that happen? So when we're going from green morph to red morph, it starts to happen on the actual aphid itself. And so what I say, the actual aphid, we get the color change starts happening, and you can get color change within 7 days. So within a week, you can go from green to red. And all of the offspring that come out of that initially green mother aphid are kind of an orangey reddish. So that happens really fast. And we noticed that the effect from the red ones to the green ones actually took a whole lot longer. And so it was more like 60 to 70 days to go from green to red. And at that point, to go from red to green. At that point, it's not the initial mother aphid that turns colors, but her offspring just slowly lose their red and go to green. And so we had a low light chamber, and that's where I was keeping my red English grain aphids. And a couple of months later, we had nothing but green aphids. And I said, well, where'd all my red ones go? Did the green ones just take over the population? But then when we put them into a high light intensity chamber, boom, 7 days later, we got the red ones back from that initial population. So they went from red to green and then back to red again. And so we played around with those two chambers and just transforming them back and forth and put it into a paper. So good question. Thank you.
Okay. So do we have any other questions? Seems that those were pretty much all the questions we had. So thank you once again, Tyler and Patricia. And
thank you for organizing.
And thank you everyone for showing up today. I know that it's pretty close to the holidays, so we kind of like want to just like log out of pretty much everything. We'll be coming back on January the 22nd, I believe, and we'll be switching to kissing bugs and ticks. We'll take a break from insect, from pretty much agricultural pests, and we'll be switching to kissing bugs and ticks. So thank you so much, and yeah, I guess we are done for today.
Thanks a lot.
So you're welcome, and you are free to log out.
So Berenice, did I come through all right while you're still there? Or did I max headroom out a lot?
No, it was actually really, really good.
That is unexpected. I wonder what changed.
I have no idea, but it seems that every time we have a seminar, I have a panic attack and somebody is struggling with technology.
Yeah, well, I had my own little panic attack too.
Yeah.
But I'm glad we got through it, and sorry that we're way over time too.
Yeah. Okay.
So I'm going to get moving. So awesome to present to you, and thanks for asking me. See you guys. And congratulations, Dr. Blake. I see you on there. Have you figured out a postdoc yet? Send us an email. Hey, bye.
Bye, Tyler.